So thanks for the invitation. It's nice to be here.
Yeah.
So the talk today is related to modeling for respiratory viral infections using fluid mechanics. But in general, my group deals with the application of all the three branches of fluid mechanics: theory, computations, experiments in different kinds of biophysical transport. So today I'm going to talk about the recent results that we have on respiratory viral infections. Now, fluid mechanics plays a key role in almost every aspect of this kind of transmissions, starting from the generation of the droplets, their exhalation, and their inhalation that can lead to infection spread. And secondly, when we think about sending targeted drug molecules to the infection sites, we need to think about the fluid mechanics as well. Now, before I go any further, let me just add this statement over here that we are not going to distinguish between droplets and aerosols in terms of the nomenclature. And I'm going to use the term droplets to imply to all liquid particulates. Okay, so the first part of the talk is on the mechanics of infection onset. And the work that I'm going to show today, it started in 2020. We used SARS-CoV-2 transmission as an example, but as I hope you will see that the modeling framework could be extended to other airborne pathogens as well. And hopefully the data that we have obtained or the modeling framework could be used in future to explain future pandemics. Okay, so before we go into the modeling, let's look at what we know about the biological mechanism of the infection onset process for SARS-CoV-2. We know that the infection starts at the ciliated epithelial cells that line the upper nasal airway. These cells, they have a surface receptor called angiotensin converting enzyme 2 or ACE2, which the virus can bind to intrude into the cells. But there is also the observation that the frontal parts of the nose has a relatively thick layer of mucus, which provides some protection against viral invasion and infection. So it was eventually concluded in a number of papers back in 2020 that the initial hotspot for infection is the nasopharynx, which, as we know, especially in this audience, is the region where the two sides of the airway would converge and is colored red in this city-derived geometries. So with this biological background, let's look at the two main questions which I posed for my group in 2020. Question one, if you look at the top ribbon, question one is, what are the hazardous droplet sizes that are mainly responsible for starting the infection? So that's Q1. Question two is, what is the infectious dose? As in, what is the minimum number of virions or virus particles that can trigger an infection in an exposed individual? So the idea was to first know the biology and then try to use our background in fluid mechanics to answer these two very specific questions. So first, I'm going to go into the first question. For this, I used those CT reconstructed geometries to first develop the simulations for the inhaled airflow. And as you can see, these color maps are for a range of inhalation rates from 15 to 85 liters per minute. Basically, I'm trying to cover a wide range of breathing conditions from relaxed, steady breathing to forceful inhalation. And these are all ADS simulations. And against that simulated ambient airflow field, we have tracked particles or droplets of a wide range of sizes, specifically from 0.1 to 30 microns. So these are droplets which are supposed to be expelled by a host. Those droplets go out in the air, they undergo environmental dehydration, and now they are being inhaled by the exposed individual. So the density, the material density should be higher than water, because when they're exhaled, it is made of oral fluids, mostly water, but as the water is evaporated, the proteins stay back and other constituents. And the density was assumed to be, based on experimental measurements from other papers, 1.3 gram per ml. And that is what we simulated for the particles. We stopped our simulation for 30 microns because beyond that, the particles or the droplets would be quite big and they would undergo prompt gravitational sedimentation. Now, within this size range, we saw that in this color map, nasopharyngeal delivery is what we are trying to track. and we see this bright hot spot, and we try to track the particle sizes for which we see that. Typically, that size range is between, I have the exact numbers here, but let's assume that it is 2 to 20 microns. So those are the particles or droplets that are most efficient at directly landing at the nasopharynx by inhalation. And hence, we are proposing that these are the droplets that we need to be primarily worried about. Now, while I was wrapping up this work, there was another conjecture that was going around that maybe environmental conditions do have a huge role to play. So we decided to look at that effect. So in humid countries, there will be less environmental dehydration. So we used just the water density for the track droplets. We didn't see that much of a change in terms of the particle sizes that are most efficient at directly reaching the nasopharynx. It was just 3 to 20 microns. And as we eventually saw through the course of 2020, 2021, almost every big country had major outbreaks at some point or another. So this answers the first part of the question, what are the bad droplet sizes? So approximately 2 to 20 microns. When we were doing this work, we didn't have any experimental validation for the work because primarily we do run some simple experiments, but I'm a bad experimentalist. I don't know how to run sophisticated experiments. So, and the groups I collaborate with, they don't have that much of a biology background to run experiments where they can study the infection onset process. So we did not have any experimental validation of the model, but thankfully, as you will see later on, some of the estimates that we have been validated in literature. So in absence of validation, I tried to look at theoretical fluid mechanics in specific scaling analysis. So we tried to develop a scaling best relationship, which can sort of predict the size ranges that we are seeing. So if you look at the Stokes number, which is the ratio between the local transient inertia over viscosity, and if all other physical and morphological parameters of this transport system, they stay the same, we can show that the density, the ratio of the density of the droplets should be equal to the inverse square ratio of the diameters, provided they are ending up at the same destination, which in this case, it's a nasopharynx. So in the last two slides, we have looked at two different densities, 1.3 and 1 gram per ml. So we plug in those numbers for the two densities on the left-hand side. And we know from the first set of CFT that the bad size range was 2.5 to 19. We plug in those numbers, and in turn, we get the bad size range for the lighter droplets. And it comes out from this scaling law to be 2.9 to 21.7, which is pretty close to what we saw from the CFT estimates, 3 to 20. I'm not saying that this is a very rigorous validation. It's just a zero-level validation of our approach. So hopefully that answered, at least to some level of satisfaction, the first question I posed. What are the bad droplet sizes we need to be worried about for infection onset? Let's move on to the next question. What is the infectious dose? That is, how many variants can start the infection? For this, again, I first started to mine through literature. I found this paper that came out in late 2020. They experimentally measured the probability of finding a variant in a droplet of a certain size. These are all undehydrated droplets. They have not lost the water yet. Some striking numbers, for example, in a 10 micron undehydrated droplet, the probability to find at least one variant is just 0.37%. So I decided to develop a simple mathematical model which hopefully can test these numbers. And to develop the model, we also needed to know what is the viral load density in this oral fluids. So that very first figure, it came from a group in Germany. They ran some tests in hospitalized patients, Munich hospital. So there is that many number of variants in 1 ml of oral fluid. We can easily figure out how many would be there in a droplet of D micron that is made of that oral fluid liquid constituents. And if that number is this n, then in other words, to come across one variant in droplets that are of this size, we need to check 1 over this caps n number of droplets. So let's think of that in probability terms. So the probability to find one variant in a droplet of D micron diameter is this P, which is 1 over small n times 100%. And this small n does contain the symbol D, the diameter. We can plug in that the value of D, and we come up with numbers that are almost similar to what we saw in the literature. So we now know that how many virus particles would be carried by these droplets. To give you a sense, if we are thinking about a 5-micron droplet, which is now dehydrated, is being inhaled, we know that the 5 micron is within that bad size range. That 5-micron dehydrated droplet would carry the viral load for a 16-micron undehydrated droplet. So we need to add up that diameter number accordingly. Okay, so with this model checking out with other findings in literature, let's now think about the actual transmission process of virus. We do need to look at what is the size distribution of respiratory ejecta out there. And there has been a lot of work over the last, I would say, 40 years on this, and there is still new work going on. But this is a paper that we used from early 2000s. They had the size distribution for respiratory ejecta. And from the CFD simulations, which I showed at the beginning, we can figure out which of these droplets and how many of this are going to end up at the nasopharynx. And once we know that, then from the mathematical model from the previous 2 slides, we can figure out what is the viral load that would be carried by these droplets. So with that connective modeling framework, we also need to know what is the exposure duration we need to be worried about, for which we looked at papers that reported the super spreading incidence, especially in Europe and in the US. A typical exposure time was 2.5 hours, when a single host managed to invite many others in a group. So if we think about 2.5 hours as the delta T that can spread infection quite efficiently, let's look at, from our CFD estimates, we can figure out the transmission rate. So at the very center, those are some numbers. So for example, if the host has a mean viral load in the sputum, for 5 minutes of exposure, the exposed individual would have 11 variants landing at the nasopharynx from our in silico data. And if the host has a peak viral load, then around 4,000 variants would land at the nasopharynx of the exposed subject. So with that rate and with this delta T 2.5 hours, we can easily figure out what is the total number of variants that is certain to start the infection. It is on the scale of hundreds. The exact number is around 300, which is remarkable. It is a very low order of magnitude. For example, if you think about the flu virus, it is around 2,000 to 3,000, the infectious dose. In other words, SARS pathogen is around 10 times more contagious than influenza A. And as you can see from some of these papers, and there are a few others which I have not added here, this number, the scale of this number on the order of hundreds has been checked by other groups who ran experiments that we cannot run in my group. Okay, so this modeling framework, I tried to extend that to other pathogens. For example, the SARS-1 virus, for 10% and 50% adverse responses, the infectious dose is 43 and 280 respectively. So very similar in order to what we are predicting. We can also possibly extend this to other viruses. For example, anisotropic viruses like smallpox, monkeypox, their airborne transmission could be a potent route. And we can apply this same tools over there. In fact, in my group, we are currently working on a project for smallpox. It's just that the target sites of the initial trigger zones for infection would be different than what we saw in the previous case, which was nasopharynx. Here it would be the lower airway, the oropharynx. But the modeling approach should be same. We decided to take up smallpox because there is a lot of work already there. We can try to validate our modeling approach with the data from the literature, and hopefully, if it checks out, we can think about applying this to other newer pathogens like monkeypox. Anyways, for the work on SARS-CoV-2, our paper did get peer-reviewed and published. I think, to the best of my knowledge, this is the first of its kind in terms of an in silico quantitative model for the infection onset process that can estimate the infectious dose of a pathogen. Infectious dose is, again, a very, very important virologic measure, something that we do need to know every time there is an emerging pathogen. So it's a quick way to estimate that infectious dose number before we are at a point to run the high-level experiments. So that was the first part of the talk on the infection onset mechanics. Let's move to the second part. And I think I have 30 minutes, right, for my talk? Yeah. So the second part is, again, on recent results that we have obtained on drug delivery. For this strand of my research program, the collaborations are mostly with industry. So I work with around 5 or 6 pharmaceutical companies or drug delivery device makers. This is the workflow. They come up with the molecules that they want to test, and they give me data on the physical parameters for the molecules as well as for the delivery devices, like what is the particle size distribution, what is the plume geometry, plume angle, etc. And we simulate the process, and then we try to figure out the optimal regions for better drug delivery. And then we combine those two building steps to come up with new formulations. For the talk today, I'm going to still focus on targeted delivery to nasopharynx, because for SARS-CoV-2 or for other viruses, for example, RSVs is another area that we are looking at. If we can initially treat the infection at the trigger zone, the chances of it spreading into the lower airway is minimal. For example, there is another study that I recently published where we saw that the infection transmission from the upper airway to the lower airway probably is happening through the shearing away of these boluses of virus-laden liquids from the nasopharynx. And those boluses are being ingested into the lungs, and that can lead to a fast infection spread. So, in other words, if you can treat the hotspot first, the chances of a more progressive infection might be minimized. Also, we can cut down on infection transmission to the outside. If the droplets that are being exhaled don't have pathogens, then it's good. Anyway, so the target site is still nasopharynx. There could be many drug delivery platforms, as we know. For the talk today, I'm still going to focus, or I am going to focus on nasal sprays. And the workflow is very similar. In the first part of the talk, we had these geometries. We have tested many other subjects, but since I had these visuals ready, so these are the same geometries that you saw at the beginning. We still run the airflow simulation, and then we run particle transport. But here, particles are supposed to stand in for the drug particles, and hence the physical parameters should be modulated accordingly. And also, this come out in a plume, of course. Like when we are trying to model the inhalation process, they don't have any plume-like conical injection. But here, since we are doing intranasal sprays, we need to make sure that the drug delivery parameters are replicated accordingly. Okay, so since we are talking about intranasal sprays, the main question that we wanted to address is, can we improve the performance of sprays that are already out there? So we looked at over-the-counter spray products in the US, and we're trying to figure out if there is any quick and dirty way to improve their performance. But to improve their performance over what they're doing right now, we need to know how they're being prescribed for usage. And we looked through the package instructions that come with products like Flonase and Nasacort, and this is the generic instruction that come with them. The patient is supposed to incline her or his forward a little. It's supposed to hold the spray bottle vertically upright and at a very shallow insertion depth into the nasal vestibule. We tried to adopt those parameters in this cartoon here. So the digital head is being inclined forward by 22.5 degrees. The bottle is held vertically upright, and it enters the vestibule and goes through a distance of 5 millimeters into the airspace. And we call this protocol as current use, or CU. Now we want to improve over this. So for which we are proposing this new improved use protocol, which is meant to use the inertial motion of the particles as they go out from the nozzle. There are 3 conditions that we are trying to check. Number 1 is the axis of the spray bottle should cut through the nasopharynx so as to maximize the use of the inertial motion of the particles. The axis should not cut through the septum because it is clinically unsafe. And the axis would eventually intersect through the side lateral walls, but let's try to do it as far back as possible. So those are the 3 conditions. And hence you can tell that based on the anatomy, this is a personalized direction. So the angle is not going to be same for every person, as long as we follow these 3 conditions. So this is a comparison on the CU direction, or the current use direction, and the improved use, or IU direction in our test subjects. You can see that essentially we are trying to hold the spray bottle with the IU as much horizontal as possible. And if I could show you the other angle, the frontal view, the spray axis is inclined a little towards the cheeks to make sure that it cuts through the nasopharynx eventually. So this is subject 1, this is subject 2. And these are some data that we have. The red lines are the ones for IU, the improved use. The blue lines are for the current use. As you can see, the red lines are doing much better. And these are just monodispersed particle tracking simulations. We have tested from 1 to 25, 24 microns. But we'll come to the part. There is a great relevance for the particle size distribution. I know, and most nasal sprays won't be able to achieve sizes which are 7, 8, 9 microns. But overall, we see that the sweet range for nasopharyngeal delivery is 7 to 17 microns. Maybe that information would be used in conjunction with the industry partners that we have to come up with a better design for the delivery device. But let's look at a few other things before we come to the particle size distribution consideration in connection to this work. So the IU is doing better, at least over that size range. We tried to test the robustness of the IU direction. If it is so that if the angle is changed by a little, then the delivery would drop, then it is of no use. But we part of the direction and we saw that there is a crowding of plots. So along the y-axis, again, we have the percentage of particles that are going to undergo nasopharyngeal deposition. The crowding shows that the IU direction is robust enough and hence could be useful if we clinically prescribe it to the patients. Now comes the validation, and with it comes the particle size distribution consideration in a spray shot. So we are using this kind of 3D printed models. I have it in my backpack over there, but I don't want to take it out because all of you guys have seen many of such tasks. So we used a model like this. The reason why it was designed in this way, as I said, I'm a bad experimentalist. I don't have high-level imaging platforms in my lab. I can possibly collaborate with other groups, and here might help me with that. But we designed this in such a way that if you think about the upper airway anatomy, if we place the 3D print, if we can measure the outflow volume, plain simple measurement of the volume, that is the volume that is going to wash through the nasopharyngeal walls. We just try to check the penetration, in other words. And this 3D print is, by the way, a third subject, not among the ones that we use for the CFD simulations, which is good because we don't want to generate a new finding that is true for just the subjects in our study. We want to see if it can be extended to other anatomies possibly. So this is the validation data. So on the left, you have the CFD numbers for the two subjects. It is measuring IU over CU, the improvement with IU on a log scale. The range is from 1.15 to around 3.25. So basically 100 times to around 300 times improvement with IU using the particle size distribution. So it is different than those line plots that I showed you. And if I go to the experimental data, for that third different subject, the range of improvement is between 2.14 to around 2.25 folds. So that range is within the overall range that the CFD exercise predicted, which is a good validation of the approach. Also, I was very happy because we used a different subject and still we could get an improvement. Okay, so that wraps up some of the recent findings. I have had other work in this kind of topics before, but this is the recent data that we have. I think I'm almost done. I want to focus the attention of this audience to a different tool, which is beyond computations and experiments, theoretical fluid mechanics. And the reason being, I do have my PhD background in theoretical fluid mechanics, where I did a lot of applied math. So this is connected to a problem that we were studying a few years back for drug delivery to the laryngeal granulomas that form at the glottis. I'm not going to go into the details, but essentially we figured out a size range for particles, 7 to 14 microns, that are better at landing over here, and that can potentially be used to improve drug design. But this project stayed with me. I have been looking at these flow fields for the last 2-3 years. There are some strong vortex patches where the deposition happened. The question was why are these vortices affecting those particular particle sizes? So I explored further into that region. I saw some very strong different vortex patches, in specific around 4 of them. And I also extracted the inlet-outlet dimensions of that region, the stream-wise depth, and used all this data to develop a theoretical point vortex model. So think about this section as a cross-section that cuts through the region with the strongest vortex patches. Those are models that stand in for this region. The first 3 visuals are same, as in this main vortex patch 2 is being replicated by one single point vortex with all the vorticity concentrated in there. All the vortex strength concentrated in there. Vortex strength, by the way, is the product of the vorticity over the multiplied by the patch area. Anyways, and this second variant is replicating this long vortex patch with 4 different point vortices with the strength equally distributed. The first equation is for the ambient airflow field. The second equation is to track the particle trajectories. Let's go to the summary of this work. We see that the particles at a size between 5 to 20 microns are deviated most from the streamlines owing to the presence of these vortices in the field. And hence that checks off with the size range that we have been seeing from the CFD. It's not exactly similar yet, but this hints that we can use theoretical fluid mechanics approaches to characterize this kind of biological transport systems as well, which would be great because CFD or experiments are just for a particular set of selected parameters, but theoretical models can actually widen our parameter space by a lot. Okay, so I think I have 5 more minutes. Okay, so yeah. No, I was asking if we have 5 more minutes. Yeah.
Yeah, a few more, just 1 or 2.
Okay, I'm just going to go through. Yeah, so these are some of the conclusions. Nothing more that interesting, but let me show you some of the ongoing work in my lab. So this video is from a group in Princeton. They saw that if we have a T-junction flow, particles could be trapped in vortex tubes. T-junctions happen in airways as well. So we are looking at whether the local deposition trends, the microscale mechanics could be connected to systems like that. So that is one project. Here we are trying to connect what Dr. Itu was suggesting, the connection of the macro scale transport with the micro scale transport. We are looking at the particle transport along with the mucociliary stream once they have landed. This last one is also quite interesting. We got an NSF grant for this, where we are trying to design filters that are inspired after animal airways, animals with a strong olfaction, because the complexity of the pathway, the fluid patterns that emerge there can actually help in trapping the particles. So we are designing these bio-inspired filters right now, working on scaling them up. And maybe they could be used as baffles that could be fitted on the ventilation ducts inside closed environments like a car or an aircraft cabin. These are the bottom lines for my group. Fluid mechanics plus virology would give us knowledge about the infection onset process. And with pharmacology, we would know more about the drug designs. So this is my last slide. I would like to thank my 2 PhD students. These are all the collaborators who have been part of all the topics that I try to touch upon today. And I would like to thank my funding bodies, primarily NSF and NIH. Thank you.
Thank you, Sakat, for your informative presentation. Now let's open up the floor for some questions. We have a couple of questions from attendees. The first one is, in your viral aerosol work, were you able to include humidity and evaporation effects? Can you comment on these factors?
Yeah, on the talk, I showed one slide where we tried to use physical parameters for the droplets that would portray the effect of humidity and evaporation on the droplets. We have not looked at a temporal study on if a droplet is continually undergoing dehydration. But if we just look at the dehydrated, the density for the dehydrated material for the droplets, we did not see that much of a difference in terms of which droplet or aerosol sizes can start the infection. So as of now, my answer would be no, these factors are probably not that dominant. The dominant factors would be the biology of the infection process, the viral load, how potent the variants are, those parameters.
Thank you. The second question is, would you recommend a very flat backward head tilt to improve drug deposition for typical nasal sprays?
I don't know about that. So the effect of gravity is quite important. And maybe the best person in this room could be some of the others, including Kia, to answer this question. But from the work that I have done, yes, the gravity does play a role. If you are not exhaling, that also does have a role. So there is no drag. The gravity does pull the particles to the target site. It also depends on what your target sites are going to be. So for example, if you think about CRS, for CRS, the target site is the osteometal complex. there the gravity could be useful. Now, would it be useful for nasopharynx for some of the other pathogens? I think that would require some research to figure that out. As of now, the data that you saw, they don't consider these different angles of the head. So I don't have any clear answer for you.